Good morning. I am Laura Menotti, and together with Mirko, we are from the Department of Information Engineering at the University of Padova. And today we present HERO-Genomics, which is an ontology for integrating and accessing multicenter genomic data. And then we will show you how we are using this ontology. This is a joint work with Gianmaria Silvello, also from the University of Padova, and Ivo Gutta and Manuel Reda from the National Center of Genomic Analysis here in Barcelona. So as a motivation, we all know that genomic data have become more and more important, both for diagnosis purposes and for understanding disease treatment and progression in personalized medicine. In recent years, genomic sequencing has become cheaper, so more genomic data are available, and that calls for a unified representation of genetic data and also for some data discovery protocols. In terms of representation, the Global Alliance for Genomic and Health, GA4GH, have started this effort and developed some standards like the Beacon data model and phenopackets. While for the trustworthy data discovery protocols, we have the Beacon API, and also we can think about federated data access, where you access different data from different hospitals. Genomic data are important for us because we are working at the Hereditary project. In this project, we are going to link related data sources and different data like imaging, clinical data, and genomic data, and we want to enable a unified query infrastructure. In particular, we want to develop a shared framework where we can do federated access and we can link multimodal health data across Europe and beyond. In this project, in fact, we have some partners from hospitals in Italy, we have hospitals in the Netherlands, and also in the USA. The idea is that the clinical data and all the data from the hospital will stay in the hospital, and we will develop this federated framework from which we can do a unified query in a single place that will query across different hospitals. And of course, we are going to develop the federated semantic polystore to do this efficiently. Going back to the genomic modeling, as we have seen yesterday in some other presentation, we have Beacon, which is an API specification that is designed to share phenotypic and genomic data. Beacon is fully compliant with FAIR data principle, so it can be used in our case and have these two components. So we have the framework and the models. In the framework, we have the request and response formats, and in the model, we have the representation of these biological data. So we just saw that Beacon complies with the FAIR principle. However, it is not perfect for our case for two reasons. So first, the implementation are left to the developer, meaning that all the ETL process needs to be done by the people employing Beacon. These in recent years have been mitigated by the Beacon reference implementation, where there are some tools to handle the ETL. However, in the hereditary case, the hereditary project, we would like to have a query platform where also people that are not computer scientists can query data. So we want an infrastructure that is easy to use and that all hospitals can exploit, even if they don't have this kind of expertise. Finally, Beacon have a data model in JSON format and is an API, meaning we cannot do SPARQL querying and there's no actual RDF ontology on top. And in the hereditary project, we would like to do this federated data access by exploiting an ontology-based data access, meaning that we are querying in SPARQL with a reference ontology, and then we will map everything in the databases that are in the hospitals. this comes to our paper. So in this paper, we present the hereditary ontology for genomic data, in short, HERO-Genomics. And this ontology represents genomic variation and all the data that are linked to genomics. With this ontology, we offer the same expressive capabilities as Beacon, but in this way, we can also use the Sparkle protocol and do some queries on it. So this is the full ontology. I know it is little, but we'll see it in more details later. Some key features of this ontology are that the terminology that we use is unified with Beacon, meaning we are using the same terminology. We also link to some ontological concepts from the semantic science integrated ontology, the gene ontology, the experimental factor ontology. So everything that could be linked to already used semantic terms has been done. The ontology is compliant with the OBDA principles. It is available online. You will see a link later. And we designed this ontology following some patterns to provide some consistency in the whole ontology. And of course, this ontology in our use case is used to do ontology-based data access for federated querying. Just to provide you a bit of context, this ontology is part of the hereditary ontology. The hereditary ontology is the backbone of the semantic data integration platform that we just mentioned. And in the whole ontology, we will describe clinical data, medical imaging, genomic data. So it will become a multimodal ontology when we put all the pieces together. The ontology as of right now consists of two parts. We have the clinical part with phenoclinical data and HERO-Genomics, which is what we are presenting today. Again, for context, the ontology will go in the semantic layer that I highlighted in red. So this is the framework that we developed. You need to look at it from the top down. So the end user will have this visualization and integration system where the user can query our system and can ask for clinical data, for genomic and whatever. For now it is on Sparkle, but the idea is that in the future you can query it for either natural language or with some specific patterns. So also people that are not familiar with Sparkle can query it. The goal of this integration, of course, is to harmonize and link heterogeneous data sources. And you see here we have the ontology-based data access, that is the layer on top. You see we have the Sparkle endpoint where we have the ontology, and then, of course, at the back you have all the federated part that we are developing. And you can also note that all the hospitals and data will be in different format and in different places. So we are not bringing the data in a unified place also for preserving privacy, but we want to keep the data at the hospitals as possible. So concerning HERO-Genomics, the domain requirements. So HERO-Genomics represent genomic variations that are usually stored in variant call format. So how many of you are familiar with the VCF files? Okay, so maybe I will just go very quickly. So VCF files have these stores genetic variation like SNPs, and the format we have the position of the genomic, the variation, sorry, the different bases that are variated, and a lot of other information. So these will all be stored in our ontology. As of methodology, in HERO-Genomics, we applied a co-design approach where we iteratively discuss with medical partners and, of course, with CNAG that are also in this paper for representing this ontology and define this concept. And we had this iterative process where all intermediate versions were submitted to them, and before moving on, we would have approval from them to guarantee that we were actually expressing the semantic of the use case. And of course, we followed some basic principle in all the ontology. For instance, we follow the when we were referencing the external sources, we use the same properties in all the ontology. We defined some naming convention, and we use the SCOS data model for classification schemes. So this is the part on genomic variation, so a small part of the graph, but you saw that there's a lot. So as we said before, HERO-Genomics adhere to the beacon data model, and genomic variation, we will talk later about the type of variations that we store, but for each genomic variation, we can link the specific VCF record and all the information from the VCF. We can link the case-relevant variants like analysis, the type of run, the zygosity of the variation, and also the biosample that was used to where we detected this variation. And also we can link the patient and the cohort and stuff, and some molecular attribute or some annotation that can be done in the VCF files. Concerning variations, we can store systematic variations like copy number variations and genotypes, molecular variations like haplotype and alleles, and legacy variation where we can store, for instance, the SNPs. And here, if you want, you have the link where you can find the turtle file for the ontology and the complete documentation with all the information on each class. So now Mirko will go on with the part on the ontology deployment.
Thank you, Laura. So as she mentioned before, in Hereditary, we have this framework in which we do not deduplicate or copy data from the data sources provided by partners to our infrastructure. That's why we exploit what is called, and we deploy our ontology in a framework that's called Ontology-Based Data Access, a well-known paradigm in the literature. And what happens in our first experimental architecture is that a user submits a Sparkle query to a Sparkle endpoint. The query obviously is based on the dictionary provided by HERO-Genomics. Then an OBDA layer exploits both the ontology and R2RML mappings to unfold and rewrite a Sparkle query into an SQL query. The SQL query then is submitted to a data federation layer that virtualizes under a virtual common relational schema all the sources that are going to be federated into the consortium. The SQL query then is submitted with the structured independent SQL query to all the sources. And then the data is aggregated back at the data federation level and returned to the OBDA layer. Here we can see an example of what happens in the reality. We have a Sparkle query, in this case, a translation of a beacon sequence query. The query then is rewritten and unfolded into SQL language, query language, from a set of mapping. Here you have an example of one mapping that has a target in triples, so that covers a piece of the HERO-Genomics ontology, and its equivalent SQL source. What happens here is that each field that is selected from the source is mapped back to the values, to the placeholder in the brackets within the target. And the translated unfolded SQL query then is submitted to the data federation and virtualization layer that retrieves on the fly streamingly the data from the data sources and recombines the results to feed back the OBDA layer. For our first experimental setup, we used data coming from the common infrastructure for national cohorts in Europe, Canada, and Africa, namely the CINECA project. We fetched the VCF files from them. And to be fair, then we will see that we used also VCF coming from the 1000 Genomes project. And as you saw in the schema, in architectural schema, we employed software as Dremio for virtualizing the data, virtualizing and federating the data sources. GraphDB as the Sparkle endpoint, and we used in particular virtual knowledge graphs repositories using on top. And for our experimental setup, again, we used the PostgreSQL NAS repositories and NAS repositories with Apache Parquet columnar database files and JSON-based database files. And in this way, we highlighted how heterogeneous data sources can be federated under a common relational viewpoint. Here also we developed a small demo with web demo. Then you will see the link to access it, in which we showcase how we can execute a variant-based query of Beacon in a semantic fashion, retrieving data from heterogeneous sources.
Okay, so just to wrap up, the contribution of this work are the HERO-Genomic Ontology, with which we can represent genomic variation and genomic data in general. And here you have the link for the ontology and its documentation. Then we also showed how we are using the ontology. So we saw that the ontology is a data model that, of course, is useful, but we also showed how this ontology is employed in practice. And for this, we have deployed the ontology in a federated context where we are using the ontology-based data access paradigm to query genomic data and data sets that are distributed across different centers. And finally, we created this small web interface to execute this federated genomic variation query. And the web application can be accessed following the link there. And also you have all the source code, both for the front end of the interface and also all the mappings that were used in the federated framework. So if you want to deploy something similar, you can start from here. Bear in mind that these projects, so the Hereditary project and all these efforts started last year. So this is an initial step. And we are open to discuss the ontology and maybe some modeling choices with clinical partners or whoever wants to get involved. And of course, also the federated paradigm. This is like a proof of concept to see that we can actually do it, but we are constantly developing it and trying to build a strong and reliable infrastructure as possible. So, okay, thank you.
Nice, this camera is right in the middle. So thanks a lot for your wonderful presentation. I really like this work. There's one comment in the beginning of your presentation that triggered me a little bit, because I'm in the group where Barend Mons is the professor. So basically, please don't state that Beacon is fully FAIR compliant, because it isn't. And it's great, right? It's a great system and it meets a need, but it is not fully FAIR compliant. And in fact, one of the reasons I wanted to also mention this, it's often in the machine readable part. So I think actually what you have done is adding to the fairness of the Beacon specifications. So in a way, I would suggest, someone of Mark Wilkinson's group is here, Alberto, this very tall guy here, to see if you can actually do that validation with one of those, I would suggest their validation tool with and without your ontology and your ontological layers. And I predict that you will see that you've actually increased the fairness of Beacon in that sense.
Okay, so we can for sure try the validation tool. Maybe we will discuss later about them. Yeah. Thanks.
So hi, my question is regarding is looking at the generalizability of your approach, because I think it can be so this general abstraction you are introducing to access different data sources is something that can be applied to many other applications rather than only to health applications. So I'm wondering, how far are you already considering generalizing that to all, opening it to all kinds of resources? And if you're not doing that, what is required to make it general, to make it really something that for all kinds of queries you want to explore?
Okay,
with
so
your framework?
I'll start to answer then, Mirko, if you want to add something. So of course, the ontology-based data access paradigm and also the federated data access paradigm is something related more to computer science, so it can, of course, be applied to any use case. And of course, you will need another ontology, I guess, because you need to represent data. So here we are applying it to the clinical data because, of course, we are in the context of this project and that's the data that we have. But still, if we manage to build a strong enough infrastructure and do some query optimization, we can for sure build this general model that then can be a plug and play for other data sources. So I think that the most is in the ontology. So Mirko, if you want to add something.
Just to wrap it up, it scales as the ontology scales. So as broad as your ontology is, and as broad are the kind of data sources that you can virtualize under this common dictionary, let's say.